Hi. Nowadays, 13% of all deaths in the world come from mental illnesses. In their life, 1 in 3 people will experience mental disabilities. There is not even one known cure for any mental illness. And I think this is because we don't really know how the brain works, but we also like a map of it. Much like the Human Genome Project in its time, our goal is to create a map on which researchers can add their data and empower them to do so. We call it the Human Brain Cell Atlas. Science's ability to understand disease comes from its differentiation of a disease and a healthy state, and its comparison of the two. Nowadays, single-cell RNA-seq allows us to read the transcriptome, the expression of every gene in the cell, to understand the behavior and the state of these cells at the single-cell level, and to differentiate them between a disease and a healthy state. Moreover, neurobiology and neuropsychiatry have made tremendous progress. Using population-scale genome analysis, they identified variants that are specific to diseases and changed the expression of some genes that might really be related to how this disease happened, such as schizophrenia, Alzheimer's disease, and more. But we don't really know how these variants affect cells and which specific cells in the brain they affect. Actually, we don't even know every cell type in the human brain. So this is exactly the goal of the Human Brain Cell Atlas, to find and sequence every cell type in the human brain. We do this through 3 steps. The first one is to access 3 brains from healthy human donors in their early adulthood that have died from accidental diseases, non-neurologically related. We then separate these tissues into smaller and smaller samples until they can be sent to single-cell RNA-seq. However, we cannot do this alone. This amounts to 1 billion cells and terabytes of data. In order to do this, we need to create a community. So the second step is to find the legal and community framework to create that and share the data, these samples with them, so that they can be sequenced in many different labs across the world, much like the Human Genome Project did. However, despite our best efforts, we will not be able to generate this data without batch effects due to small differences between these labs and the sequencers. So the third goal is to create an algorithm that can look at the different expressions among cells and find the variability that relates to the cell state and the variability that relates to the batch effects, to remove that one and create a one seamless human brain cell atlas. I think the ability to create this human cell atlas might transform and empower the neurobiology and neuropsychiatry fields. It might be a key resource for researchers to add their own data and compare healthy states to disease states and predict which gene and which cell types these variants that they created affect. To end with this, the Human Brain Cell Atlas might become what the Human Genome Atlas was at its beginning. Thank you very much.